So chapter 21 is about blood. This is still week 2 content. This can still be on our week 3 exam. It's just blood. We're not doing the entire cardiovascular system. We're actually going to separate the heart and blood vessels after this exam. So we're just going to do the general composition and functions of blood, what is in blood plasma, the fluid portion of blood, the formed elements that are within the blood. There are 5 white blood cells, and then you have your red blood cells, and also you have platelets. Those are formed elements. We'll go over blood typing, and there's a little bit of a hurdle there where we're going to differentiate antigens and antibodies. Antigens being found on red blood cells and antibodies being found in serum. We'll talk about hemostasis, what happens when you slice into a blood vessel. How is that going to clot? And then how is that clot going to retract and go away without embolizing and killing you? We'll talk briefly about disorders of hemostasis. We'll talk about hemopoiesis, which is the production of blood cells. Erythropoiesis is specific to red blood cells. And then a couple of erythrocyte disorders. It is a little bit patho-content, but we will cover it here today. So whole blood is made of red blood cells, white blood cells known as leukocytes, and those are going to be found in a place called the Buffy coat. Platelets, also formed elements, and then the fluid known as plasma. So if you take a sample of your blood, you put it in a test tube such as this, and then you centrifuge it, the heavier portions are going to fall towards the bottom of the tube, and the lighter portions are going to stay at the top of the tube. And this is known as the hematocrit. And you do need to know the word hematocrit, and you need to know the percentages. So hematocrit means that about 45% of your blood sample should be formed elements. 44% should be red blood cells, and 1% should be a combination of every type of white blood cell and platelets. The remaining 55% should be plasma. So what's the hematocrit? What's the percentage for the hematocrit? 45% is the percentage for the hematocrit. In a healthy person, that hematocrit should be 45%. That's how we say that. The function of the blood is going to be transporting gases and nutrients and wastes. So we're going to talk about gas exchange when we get to respiratory. We're going to talk about nutrient exchange when we get to digestive. And we'll talk about wastes, especially when we get to urinary. The way that these places get to, or these substances get to these systems is via the cardiovascular system through the blood. It's also going to help you regulate things like your temperature and pH. So temperature via things like vasodilation. If you vasodilate near the skin surface, you can radiate a lot of your body's heat. Whereas if you vasoconstrict near the surface, you conserve more of your body's heat. pH is going to be your acid-base balance. Clinically, one day that's going to be very, very, very significant to you. And your fluid levels and fluid shift, fluid balance regulated through your blood, and all of the things that filter your blood, also very, very clinically significant. I actually have been working with Karen this quarter for our quarter 4 content, and she actually has about 3 to 4 weeks just about fluids and fluid balance and IVs and how we can alter and affect that and improve it. Protection, those white blood cells, leukocytes, are going to be our immune system. And that blood carries those antibodies. Those antibodies, among other things, will be secreted by some specific white blood cells. All right, we do definitely need to talk about the proteins found in plasma. This is one of the most important concepts you're going to see today. Not the only one, the most important. I want you to never forget the word albumin. Albumin is the first plasma protein we're going to learn about. Albumin is going to be stuff. And where does water go? Towards stuff. Albumin is the stuff in your bloodstream that's going to keep the water in your bloodstream and not leaking out of your bloodstream too much. What happens if you leak too much fluid out of your bloodstream? It's not going to move forward, sure. What about, like, just think about your legs. If you've got a bunch of fluid, like, leaking out of your bloodstream into your legs, what do we call that? Edema. So albumin is going to be the thing that keeps us from being edematous, which is why this is a concept that's literally never going to go away. We're going to talk about albumin for cardiovascular system. We're going to talk about it for respiratory, digestive. It's never going to go away in every day in patho. I just covered this yesterday in patho as well. Albumin is going to exert osmotic force to keep fluid inside of the cardiovascular system. Globulins are going to be part of our immune system. They're going to be part of transporting those antibodies. Immunoglobulins is another word for antibody. And fibrinogen is going to help us clot. So again, this will be found, get my laser pointer back up, in the plasma portion. This is not a formed element, this is the fluid part. These are proteins. They are very small. They are small enough molecules that they stay suspended in this fluid, in the plasma layer, with that hematocrit test. Other things that can be found in plasma, electrolytes. Electrolyte balance is also very clinically significant. Nutrients, again, that we get from food that we eat, that we transport to tissues, respiratory gases, oxygen and carbon dioxide, and wastes, cellular byproducts that get picked up, and we're going to try to pee them out through that urinary system. Aside from quantitatively assessing blood via the hematocrit, we can also assess blood with a blood smear. And this is an example of a blood smear. This is not actually a particularly healthy blood smear. This is a blood smear that shows you a lot of white blood cells, just to differentiate them visually from the red blood cells. Red blood cells look like these biconcave discs. They look like a little bit like inner tubes to me, but you know, the inside of the inner tube is continuous. We do have to stain blood to get it to look like this. If we didn't stain it, they would all just look clear through a light microscope. So this is with an H&E stain. In a normal blood smear, there should not be this many white blood cells. This person has too many white blood cells. This is a disease state. But we can start to recognize and acknowledge that you're going to have different types of white blood cells. You're actually going to have 5 different types of white blood cells that we're going to need to cover today. This one's showing you only 3 kinds, 2 neutrophils, which is reasonable. You have more neutrophils than anything else. Lymphocyte, 1 lymphocyte, and 1 monocyte. So notice the nuclei. Before we even get into the very specifics of each of these things, notice the nucleus. This has a multi-lobed nucleus. That makes it very, I think, in my opinion, easy to distinguish from anything else. So if you see a big blobby thing like that, that's a neutrophil. That monocyte has a horseshoe-shaped nucleus, and that lymphocyte has a very large, sort of rounded nucleus with very little cytoplasm around it. So just getting you the sort of preview of how we're going to visually differentiate them, and you will be held to visually differentiating them. Also in the smear is an aggregate of platelets. When you see little individualized dots, platelets are cell fragments. So there was a progenitor cell, there was a sort of a stem cell for this, and it broke off pieces of itself, and those are platelets. And those platelets are going to help with the clotting cascade. So already, we're barely 10 minutes into this lecture. We've already seen 2 things that help with the clotting cascade. That was fibrinogen, the protein, and platelets that help form the plug. Red blood cells, or erythrocytes, the prefix erythro means red, cyte just means cell, are smaller than the average cell size. Unique biconcave shape. This shape is very carefully held together structurally by a dense cytoskeleton and by hemoglobin. Hemoglobin is actually part of the structure of the red blood cell that does have clinical significance. Part of the reason that they are so small is that they need to fit through capillary beds. And when we get to blood vessels, we'll talk about what capillaries are and how they are different from larger blood vessels. It's a very small space, and as these red blood cells march single file through capillary beds, they form something called the Rolo. And probably the candy is named after the red blood cells. I'm just going to guess that. Anybody ever had Rolos? Yep. And they look kind of biconcave, and they're all stacked. Yep. There it is. So hemoglobin is a complex protein. It's actually made of 4 protein subunits. Each of these protein subunits is known as a globin. And each globin has a heme group on it. That's this molecule right here that binds to an iron, and that iron in turn will bind to an oxygen molecule, specifically O2. So oxygen likes to float around in the form of O2, not as a single oxygen molecule. Therefore, we need iron to form hemoglobin. Without iron, we can't form hemoglobin. Without hemoglobin, we can't transport oxygen effectively in our red blood cells. Do I have a number? Every single red blood cell has 280 million hemoglobin molecules, which is crazy, right? That's your, multiply that times 4, and that's your molecular oxygen carrying capacity for every single tiny 7 micron red blood cell. So you had a question.
I answered it.
Okay. So just to give you some future insight, the fact that this requires iron means iron deficiency anemia is going to be related to the formation of hemoglobin and its oxygen carrying capacity. Does that make sense? All right, here's the birth and death of your red blood cells. Your red blood cells, all of your blood cells came from your red bone marrow, all of it. All of your red blood cells, white blood cells, platelets, all of them came from the red bone marrow. So you synthesized your red blood cells in your red bone marrow. They got transported to your cardiovascular system where they will circulate for, right there it says 120 days. As these cells actually leave the red bone marrow for the blood vessels, they lose their nucleus. And what is the significance of a nucleus? What's going to be there that's really important to cell health? Chromosomes. What's on the chromosomes? DNA. And DNA is the blueprint, right? We use the blueprints to make protein components of the cell. So as these cells degrade, in a normal cell you would have those blueprints to replenish those proteins, to improve and correct those proteins, right? In red blood cells you are lacking the capacity to replace degraded proteins. So they will have a relatively short lifespan of 120 days, at which point they will no longer be usable. Your body will have to filter them, and it will filter them in the spleen and a little bit in the liver. So that spleen is going to find those defective red blood cells, and it's going to break them down, and it's going to recycle anything it can recycle. It's a really fairly efficient recycling center. Just like any recycling center, it's not 100% efficient, but we'll worry about that when we get to lymphatic. Part of the cycle is that now that you've phagocytized your old defective red blood cells, now your red blood cell count is a little bit lower, and you're actually going to have to go back and synthesize more red blood cells. Questions so far? What was that hormone from our last lecture that helped us to synthesize more red blood cells? Nope. From the kidney. not A.
Erythropoietin.
Erythropoietin.
Yes.
Yes, erythropoietin. All right, you had a question over here.
Yeah. So if platelets are the fragments, is that fragments of like old? Okay.
They're fragments of a cell. I think we see it later in this lecture of something called megakaryocytes.
Okay.
Yep. Okay, but for now, we're going to start with blood types. Does anybody work in an environment where you do blood type testing or blood type testing is done on a regular basis? Or is anybody already really comfortable with blood type testing and cross-matching in here? No? Okay. It's a learning curve. It's a very steep learning curve, but it's a very sudden learning curve. 1 second you do not understand it at all, and the next second you understand it completely and can apply it to everything instantaneously, and it's easy. So you're going to not understand it right up until the moment that you do, and that is okay. It's going to take you different amounts of times to get there, and that is also okay. You will all get there. And I think I'm going to do a thing, if I can, if it'll let me. No, it's not going to let me. All right, I'll try to do it on here as much as possible. If I need to pause it and get a webcam up to do it on the board, I will do it there. Generally, this is a draw it on the board situation. You have to understand what an antigen is, and you have to understand what an antibody is. So I'm going to start with antigens. You're going to want to write it down. An antigen is a protein found on your plasma membrane that we can identify as self or other. What I mean by that is every cell in your body is wearing a little name tag. For me, all my cells have a little name tag that says, "Hi, I'm Reeves." Those are my cells' antigens. If a bacterium makes it into our system, it has antigens as well. And that bacterium is going to say, "Hi, I'm E. coli" on its antigen. And my body will recognize that antigen as other, and that will stimulate the immune system to destroy it. So far, so good? So antigens are the identifier. They're a protein identifier.
Where are they found again?
Plasma membrane. They are found on the plasma membrane. They will always be adhered to the cell. Give me one of these when we're good. Okay. Antibodies are going to float in serum. We talked about globulins, immunoglobulins. Those are antibodies. So they are plasma proteins, and they're going to be specific. What they are going to do is they're going to adhere to the antigens and do things to that cell, depending on if it's an identified that cell. So this antibody is an anti-B antibody. It's going to attack protein B. Antigen B. So what maybe you're trying to get, or maybe what you're trying to see here is that we really have to differentiate the term antigen from antibody. They are doing directly opposite things. The antibody is going to act against the antigen. If it finds a non-self antigen, it will attack it. Everybody's got antigens all the time. Not everybody's got antibodies. So these are opposite concepts. That's the most key part of this thing today. How are we doing so far? So far, so good? Okay. And we will talk more about what antibodies do specifically, again, when we get to that lymphatics lecture. It's going to be kind of heavy immunology to really understand everything that antibodies are going to do. We're only going to see one action of antibodies today. So here's what blood types actually are. Your blood type is what type of antigen you have on your red blood cells. If you have type A blood, then you have an antigen that we have simply named the surface antigen A. If you have type B blood, then you simply have the protein antigen that we've named surface antigen B. If you have type AB blood, you possess both of these antigens. If you have type O blood, it simply means you do not have any of these antigens. Good? So far, so good.
I'm confused just because you said everybody has antigen.
Yes, so O does not have A or B. There are other self-identifiers that are not going to play into this blood typing system. Yes. And that's a very important point that is very, very confusing for people. Yes.
But how's it like where isn't it like if you have type O blood, anyone can get that blood?
Yes, so we will have universal donors and receivers. We're not there yet. We're going to build up to that concept. Yes.
Are these on every single red blood cell?
Yes, this will be on every single red blood cell that you possess depending on your blood type. Okay, so there's the second half of that. Actually, I shouldn't say half because there's going to be more down here. For this first part, here's the second half of it. Your antibodies are going to be the opposite of your antigens. So remember, your antibodies are there to attack foreign antigens, right? They're not there to attack you. They're there to attack others, to attack foreigners. So if you have type A antigens, you cannot have the anti-A antibody. That would kill you. You would only have the anti-B antibody, but you will have the anti-B antibody. This is the first hurdle. How are we doing with this first hurdle? Okay. If you have surface antigen B, you cannot have the anti-B antibody. That would kill you. You will have the anti-A antibody, but you will have it. Yes. Yes, if you had the antibody for your antigen, it would kill you. If you have both type A and B, by definition, you can't have either of these antibodies. Either one of these antibodies would kill you. But if you are type O, you now have both antibodies. So putting either A or B antigens into this system would kill you. Or sorry, would destroy those. Okay. Next part of that. How are we doing on this so far? So far, so good. When we do blood donations, consider only, okay, jazz hands for this, write this down. Consider only that the blood cells are going. Do not consider the plasma. Consider only the blood cell. This is going to be a one-way street for donation. So when we say we're donating type A blood to this person, so we start here, we'll start in this row. This is the blood cell, and it's going into this serum. It's going to be a one-way street. And see where I've put my cursor right here if you're writing down. So we'll take this blood cell, we'll put it into this serum. We will not consider this blood cell coming in contact to this blood cell because that does not matter. We will consider this blood cell going into this serum, or if we're putting this blood cell in this person's serum. That is how we will worry about our blood donation types. Okay? One-way street, one way only.
Can you say that again? That with blood donation, consider only.
Yes, with blood donation, consider only the blood cell of the donor going into the serum of the recipient. Blood cell to blood cell means nothing. Blood cell to serum is what is going to cause our cross-match issues. Again, do not take this person's serum and put it into this person's serum. Sure, some of that serum is going. The effect is going to be small enough that we're not going to need to worry about it. We're going to worry about the cell going into the serum. And then we're going to ask ourselves if that's going to be okay. Questions?
So, plasma donation is different from blood donation.
Yes.
But if you're the blood drop, right? That thing that it takes all of it. So do they just filter out the plasma before they give?
They can, yes. So there's
So
such
why
a.
can't they use that plasma? Like, why do you have to do separate?
I'm not entirely sure what the procedures are for those, for serum donations. If anybody knows, feel free. Okay. Were there any questions before I keep going with this? Go for it.
So then, for example, I have type A blood.
Mm-hmm.
So then, in a sense, is it like the antibodies are attacking everything without that name tag A, in a sense?
Yeah. So these antibodies will be very specific. So let's say that this is Angela and this is Bob. This is anti-Bob, this is anti-Angela, right? So so let's do an example. Let's take Bob's blood and put it in Angela's serum. What's going to happen to Bob's blood? It's going to get attacked. So the anti-Bob antibodies are going to attack Bob's antigens. What they're going to do is agglutinate it. Agglutinate means to clump. So what's actually specifically happening is this binding site and this binding site are going to attach to two different red blood cells and keep them attached to each other. And there's going to be enough antibodies around that a whole bunch of red blood cells are going to be stacked and clumped. And that's a clot, right? We don't like clots. So now if we give Angela's blood cell to Bob's bloodstream, what's going to happen? The anti-Angela antibody is going to agglutinate Angela's cells. Okay, so far so good, right? Now we put Angela's blood into this bloodstream. Are we okay? Yeah, we're okay. We put Angela's blood into this bloodstream. Are we okay? Correct, we are not okay. So far so good. You guys are doing great on this. Don't be surprised if we get to the practice and you're back where you started on this. It makes sense when I explain it, and then there's another hurdle when we get to practicing it. Okay, one last bit. We had this all figured out. Blood type donations were going way, way smoother. Occasionally something would go wrong, and also sometimes babies would die. Then we discovered the RH factor. I'm not exaggerating, I'm sorry, there's babies dying, it's really sad. The RH factor is simply another protein. It's the D-type protein. We could have just called this type D if we'd wanted to. We called it RH factor because we discovered it in rhesus monkeys. That was our model organism, RHESUS, rhesus. So the RH factor is simply the presence of another protein or absence of another protein. It is the protein type D. People do not automatically develop anti-D antibodies, sorry, make sure I use the right word, anti-D antibodies until they've been exposed to the antigen type D. So the way some people got exposed to antigen type D was if mom had no surface antigen, but her baby had surface antigen D. So during embryological development, there is a blood placental barrier that separates them, but there are all kinds of things that can cause exposure between fetal and maternal blood. Needles, trauma, anything. And the birthing process, obviously, is a really easy time for you to have exposure. So here's what was happening. Mom's D negative, baby is D positive. So RH negative, RH positive is the real way to say that. First pregnancy with a fetus with the D-type protein, fine, baby's born. But mom gets exposed to the D-type antigen and develops the anti-D antibodies. Second pregnancy with a child with the antigen type D. Any exposure to that fetal blood is going to agglutinate the fetal blood. And that's why babies died. It's called erythroblastosis fetalis, or hemolytic disease of the newborn. I believe we've got a slide for it. And that is how we discovered, unfortunately, this type D. Now there's a shot for it. You get the shots, that doesn't happen. You're fine. Baby's fine. Everybody's fine. Yes?
So, when my daughter was born, they were concerned because they said, oh, she's rejecting your blood. And they were constantly doing like a blood transfusion or something. Is that what
Possibly.
Possibly what it was?
Yeah.
Okay.
Yeah.
So are there 5 types of?
So this is A and B. This is positive and negative. So we don't say the phrase is that you're RH positive or negative, but this would be AB positive if you also have the D protein.
Okay, that's fine.
And just AB negative if you do not also have the D protein.
So you have a type A, B, AB, or O, and you also are positive or negative?
Correct.
is there any combination that like is not going to happen, or is there? You
They
can
can all
have
happen.
Positive or.
Yes, you can have this in any combination. You can have type A positive or negative, type B positive or negative, type AB positive or negative, or type O positive or negative. Yes.
So they test every single woman that is pregnant if she's positive or negative.
Mhm.
If you know, like if I know that I'm B negative, would that be B and then RH? Like why would they have to test for RH blood type?
If the father is positive.
Ooh, so if I knew the father's blood type and my blood type, they wouldn't have to do this test?
Possibly if you're both negative.
Okay.
So there, it actually follows, you can do Punnett squares for this, and if you want, we can draw some Punnett squares for this later. Actually, I think we might actually have to do some Punnett squares for exam purposes. So the negative is recessive, so you need 2 copies of the negative to be negative. The positive is dominant, so you only need 1 copy of the phenotype for D to express the D protein.
100%.
Yeah, whereas these can co-dominate with Punnett squares, so if you're type AB, then one square is A, the other square is B. And then if it's type A, let's see, type O is recessive, type A and B are both dominant. So if in order to have type O, both mom and dad have to be type O.
Could fraternal twins, like one be positive and be negative, and both survive?
I mean, with a Rogan shot, yeah, definitely. Yeah, I believe so.
So who gets the shot? The mom?
Your mom gets the shot.
Okay, not the baby, like that's it.
No, I think it's just a maternal injection. Has anybody had the Rogan shot? No? Usually there's like 3 people. Like, okay. All right.
So that would change. So if you got the Rogan shot, so like if you were A negative, you would then be A positive.
No, it doesn't change your blood type. It
So
just
you don't produce those
it just keeps you from
antibodies.
prevents you from producing antibodies. Correct. Yes.
So then, for example, I'm A positive. So then how does the antibodies know not to destroy my blood cells when I have, in a sense, 2 genotypes?
So if you're, what did you say, AB positive
Yeah,
or A
no,
positive?
I'm A positive.
So if you are A positive, which antibodies will you have in your serum? And you can figure this out. I'll do this on the board. I'm sorry for my listeners at home that you don't get to see this. So here's your cell. You are, let's see, it's doing A's as circles and positive is a square. So what's your serum going to look like?
B
Anti-B antibodies only. So you're only going to have the opposite antibody to the antigens that you have. You will never have the same antibody as your antigens. If you did, you'd be dead.
Yeah, I understand that part, but I'm saying, like, since now I have the square,
Mm-hmm.
in a sense, I have 2 name tags, so, okay, it only sees the 1 name tag, but it also sees the second.
There are no antibodies
are
for.
the triangles. So that
Yeah.
doesn't fit either the circle or the square, so it's not like that.
There's no square antibodies.
Right.
Oh, okay.
Yeah.
Okay, let's do a different one. So there's a triangle now. And potentially, let's just imagine every potential antibody that you can now have. What antibodies could you potentially have? Anti-A. And you'd want to say anti-A and not A antibody, just to be a habitual thing. Okay? Anti-A antibody, and potentially what else?
Anti-B.
Anti-D.
Wait.
Because if we had anti-B,
It would be back to triangles.
Right.
yeah. So what would happen if you had that situation?
Is that the autoimmune?
Clotting. Kind of. So antibodies and autoimmune diseases, yes, related.
Okay.
This, blood cells, not so much.
Okay.
Not for our purposes.
Okay.
Yep. Okay, so you will never have the antibodies for your own antigens. You will have antibodies for every other antigen, potentially. And we just assume the anti-D antigen. Whether or not there's previous exposure, we just assume it. We're not going to risk it.
Until you're tested and you know you don't have it.
For blood type cross-matching
Okay.
purposes, we're going to assume they have that. So for example, if I needed to donate blood to this person, and they may or may not have developed this antibody, but I am positive, we'll just say, no, we're not going to have you donate, just in case. Okay, so let's now create a donation scenario.
So is that why A or O negative is a universal donor?
We're getting to that, yes.
Okay.
Yeah. We're still getting there. I wanted it to be step by
Sorry.
step.
Okay.
If I lay it all out at one time, you won't work. So, so let's do that. Let's do type O. O negative. Here's your red blood cell, nothing on it. What do we consider for donation purposes? Do we say we put the blood cell in the serum or we put the serum in the blood cell? Blood cell in the serum. So this blood cell, what are we, if let's say we put this here. Are we okay with that?
Mm-hmm.
Yeah, this is fine. So type O negative can donate to what's this blood type right here?
A positive.
A positive. O negative can donate to A positive. Okay, put this blood cell here. Is that okay?
No, because the negative?
It's okay. Why is it okay?
Because it also has a serum, but there is no.
There are no surface antigens on the type O negative blood cell. Therefore, you can put this in any scenario. You can even put it in another O negative. That's fine. Right? So this is going to have all three. But doesn't matter, does it? Because this has no surface antigens that can be used to agglutinate. Cool, right? So that's why O neg, universal donor. Makes it easy. Okay, let's reverse the situation. Let's make it, we're going to make two things. AB positive. Okay. Put this here. What happens? Right. The anti-B antibody attacks the B antigen. And this agglutinates. So we cannot put AB positive blood in an A positive person.
Would the anti-D also attack it because they both have?
Is this going to have anti-D?
Oh, no, because it already has it, sorry.
Correct.
You can't do that.
No. You cannot put AB positive blood in an B negative person because there's the potential for both the anti-A antibody and anti-D potentially antibody to attack this AB positive blood cell and agglutinate it. Does this person have any antibodies? No. They have every antigen. They are also AB positive. So if you put this here,
That's okay.
yeah, we're fine. Check. So AB positive can only donate to AB positive, but reverse this. Let's say we were donating A positive to AB positive. Yeah, we're fine. B negative. Yeah, we're fine. AB positive. We're good. AB positive is the universal recipient. They can barely give to anybody except for themselves. They can take from everybody. They're the selfish jerks of the world. AB positive is selfish. O negative is selfless. But if you're O negative, again, look at your antibodies. Which antibodies do you have if you're O neg? A and B. Anything else? And potentially anti-D as well. You're rocking every single antibody when you are O negative. So let's try to give this to O neg. Nope. That to O neg. They can't receive from anybody except for O neg. So they're the universal donor. They cannot receive. AB is the universal receiver. They can't donate. It's not that they can't donate. They can only donate to their own types or receive from their own types. Yes?
Are there blood types that are more likely to get certain diseases?
Arguably, yes. actually, yes.
Which one?
I'm not as familiar with that, unfortunately. But I have heard that evolutionarily there are some blood types that are more common in certain areas than others. There are predispositions based on those things. They're subtle, though. It's not a major effect, as far as I understand it. Okay, so you've done some of this here. You'll have practice on your worksheet. Again, if you cross-match incorrectly, the reaction is agglutination, and that does lead to clumping. Clumping is literally the definition of agglutination. The clumping is caused by the binding of these antibodies to consecutive cells, and there's enough of them that many, many cells will clump together. And in our blood type testing, that is the reaction we use to determine blood types. So this is what it looks like. This is non-agglutinated blood. There was no reaction here. This is agglutinated blood. If I ever get your lab open today, then this is what we will get to see with our blood type testing. It might not happen until next week, sorry. All right. How are we feeling about that concept right now? Pretty good? Okay. Again, the key to this is take the blood cell, put it in the serum, not the other way around. And we'll look a little bit more at blood type testing in a second. Okay. The results of agglutination: diminished oxygen carrying capacity, diminished blood flow beyond blocked vessels, and that hemoglobin from the destruction of some of those red blood cells ends up in your kidneys and can lead to renal failure. A bunch of protein all at once in your kidneys is not a good thing. And here's what that's going to look like as a transfusion reaction. As future healthcare practitioners, these are the symptoms to watch for after you've given some blood. Yes?
I mean, it's probably a very immediate reaction because it's all over the blood, but like how quickly would it take?
I'm not sure. I imagine it would be pretty quick, like you say. Do you have any insight into this? Are you awake?
I'm awake.
Okay.
Yeah, not really. I mean, not that you haven't covered.
Okay. Anything for speed at which a transfusion cross-match would have that reaction?
That I don't know, because
No.
we don't do anything for it.
Okay.
Products in the field.
Gotcha. Good to know.
It's pretty neat, like, we ever give, it's like when you give blood, you just, like your blood pressure lowers, that's why they give you snacks and
Yeah.
juice afterwards.
Yeah. It's different from a cross-match reaction, though. It's just low blood pressure. Yeah.
Is it lifetime dialysis or just once?
I imagine the dialysis would probably be one time unless permanent kidney damage was caused.
Okay, so unless it was that total overload.
Again, ask a nurse. Not a nurse. Okay, back to erythroblastosis fetalis again. Rh negative mom, Rh positive fetus. Mom had to be exposed once, and it's the second pregnancy that is affected. Okay, white blood cells. On to the next concept. And this will probably take more time than we need before we go on break, so we'll just do an overview, and then we'll go on break, and we'll go into detail on your white blood cells. White blood cells are pretty cool. There's 5 major types of white blood cells. They're capable of, this means walking and moving towards chemical attractants. So diapedesis means they're going to roll along in your bloodstream until they are needed elsewhere, and then they will travel. They will travel towards that chemical attractant. It's not by will. I like to anthropomorphize things and say they want to do this, they go to this place. It's all chemically based. These are biochemical reactions that we have pretty much figured out at this point. So diapedesis and chemotaxis, those are just the sort of biochemical explanations for how they walk and how they are chemically attracted to locations where they're needed. You have 2 classes or types of leukocytes, granulocytes and agranulocytes. Granulocytes have granules, agranulocytes don't. That's about it. It just helps you classify them by appearance, slightly by function, but they're all going to have slightly different functions. So here are your 5 main leukocytes, your white blood cells. They are all super cool. They all have names and personalities. I love them all. This is not a searchable page, but this is an important page, so if you've got your PowerPoint open, maybe make this searchable for the roles of white blood cells. Otherwise, give it a star, say this is the slide you need for the roles of white blood cells. And it's a pretty general overview right now. Now I'm going to go in order from most frequent to least frequent, which is not the order they are in on this slide. We're going to start with neutrophils. Oh, I should tell you, there's a mnemonic device for remembering frequency, and it is never let monkeys eat bananas. That's going to help you remember frequency. So we're starting with N. We'll start with never. Never let monkeys eat bananas. Start with a neutrophil. Neutrophil is the most common white blood cell. It is a granulocyte, which means it does have granules. And it has this multi-lobed, weirdly shaped nucleus. It has two major mechanisms. It's going to phagocytize, and it's going to release enzymes that break things down. So this is your first responder. This is the guy that shows up when you cut your arm on a tree branch. When you get pus, if that starts to get infected, that white stuff, that's neutrophils. That's how many you have. You have a lot of neutrophils. The white stuff from pus in your zit, that's neutrophils. So a bunch of them arrive, and they're there to clean up. They're there to attack and consume any bacteria that show up in that cut on your arm. They are there to break down any cellular wastes from damaged cells from that cut in your arm. They're going to clean up, they're going to respond, they're going to try to keep you from getting infected in the first place, and if they can't, they're going to keep swarming until they've made it all pussy and gross. That's your neutrophils. So again, the important part for that is phagocytosis and lytic enzymes. Never let monkeys eat bananas. Lymphocytes are next up. The key words for this are specific and innate immunity. When we get to our lymphatic lecture, we are going to spend a lot of time on even more subtypes of lymphocytes. Some of them are going to work in innate immunity. Some of them are going to act in specific immunity. These are the ones that are going to go to the dojo and train to kill specific things. So again, keywords, innate and specific immunity. That's all you really need to do for now. We will learn a lot more about these guys later on. Never let monkeys. Next is monocytes. Monocytes are macrophages, which means they are big eaters. They will help break things down. Never let monkeys eat. Next is eosinophils, my personal favorite. I love these guys. First of all, because look at them, they're pink and purple. They're always pink and purple like that. They have these granules that stain this very bright pink, and there are very, very many of them. The thing that I like about eosinophils is that they help us kill parasites. Yay! And later on I can show you guys a video of some eosinophils killing a parasite. They do it somewhat similar to the way that neutrophils do it. What they're doing is they are going to either try to eat them as a whole, phagocytosis, or release some more lytic enzymes, just like that neutrophil is. The difference here is that they're going to be a little bit more specific to those parasites, and they're not nearly as frequent, right? Hopefully parasites aren't that big of a concern for us in a first world nation. Fingers crossed, knock on wood. I hate parasites. I have a totally irrational fear of them, but so these beautiful pink and purple guys are going to help us kill those. Finally is basophils. Never let monkeys eat bananas. Basophils are the least frequent, thank goodness, because they're the jerks responsible for all of our allergies. They're, again, granulocytes, and those granules contain, among other things, histamine. And histamine is great for causing a lot of inflammation, and it's great for responding to some pollen that was never going to hurt you in the first place. Yeah, that's what they do. They also help with parasites, so I can't really hate them that much. But obviously this is going to be a major target for your allergy medications.
So if you have severe allergies, is that you have a lot of the basophils? Potentially.
If you have severe allergies, potentially it's related to basophil frequency. It's probably going to be a lot of other things too. So this is not the only allergy cascade. There's a bunch of other cells related, like mast cell formation and about 20 other things that we can target with allergy medications. But we're simplifying. Okay, those are your 5 white blood cells, and I think it is now time for a break.